Hello Giulia.
Bonjour Christophe.
Bonjour.
Je sais pas si Giulia est déjà là, mais il est 8 heures, c'est ça? Ah non, moins que ça encore. Il est beaucoup plus tôt.
Ça, il est 8 heures du matin.
8 heures du matin, d'accord.
Et comme je dois donner une présentation juste après, du coup, je suis déjà sur le lieu de travail.
D'accord. Hmm.
Hi everyone, can you hear me?
Yes.
Yeah, very well.
Okay, just 1 second. We have some delay. There were problems with the jury, but they are coming soon.
Sure.
Okay, good.
By the way, Julia, I have a bit more time because Habib pushed my talk at 11:30.
Okay,
We
nice.
are, and it's just one floor up from here, so I can, so yeah, we have, for you, it's until 11:30, so yeah.
It's a good coincidence since we
11,
are a bit late.
11:20, I would say.
Okay, thanks.
Okay.
Oui,
Ça va?
oui,
Je t'embête pas longtemps.
ouais,
J'ai une question. Dans iPad,
oui.
il y a le 3D ASL et il y a deux séquences d'ASL en 2D. On fait pas tout.
Non, il n'y a que le... Pardon, attends.
Unmute. What's the problem? You have to cut the one from your side. Ok. Vous passez.
Faut faire mute là, non?
I just leave the microphone. Okay, I will cut completely the audio. Yeah. Now it should be okay. Yeah.
C'est bon? Là, ça fait ça. Voilà.
If you go and ask Patrick,
We should cut the sound from the TV.
Yeah.
Yes.
Now, c'est maintenant.
Est-ce que tout le monde entend bien? Il n'y a pas d'écho? Tout va bien? Ok, on y va? Okay, so Giulia Rocco, we first listen to your work, and then the jury will ask a question about it. So please.
Okay, for the people on Zoom, can you correctly see my screen? Okay, we'll just move it there. Okay, great. Okay, hi everyone, I'm Giulia Rocco and I'm going to present you my work, Multimodal Functional Exploration of the Cerebellum. As you can imagine, the protagonist of this work is the cerebellum, and it's the orange part in the back of the head. It's part of the central nervous system, and together with the brainstem and the cerebrum, it forms the brain. But what makes it so special? First of all, even if it has only 10% of the brain mass, it contains 80% of the total number of neurons in the entire brain. Then, if we take the surface and we unroll it and we flatten it, it's almost 80% of the total surface of the cerebral cortex. And it's especially a stripe that is very narrow, 10 centimeters, but 1 meter long. We can see already from this video that the cerebellar cortex has a very different appearance from the brain. There are not these deep sulci and gyri, but there are a lot of circumvolutions that are very tightly packed. You can see also on the pictures on the right. Okay, so you can see my point. Yeah, on the picture on the right, that it has a tree-like structure, very ramificated, and it's called arbor vitae. Anatomically, it can be divided into two hemispheres, the left and the right hemisphere, and they are connected by the vermis. If we unroll the surface, we can divide the hemisphere in 10 lobuli, and these lobuli are constructed by following the main anatomical features, as you can see here. The biggest one are crus 1 and crus 2, and all the other ones are designated with a Roman number. For a long time, people thought that the cerebellum was considered to be involved only in motor control. However, recently there was a neurologist at the Massachusetts General Hospital, Dr. Schmaman, who had patients with extended cerebellar lesions. And besides exhibiting motor symptoms, they also exhibited deficits in executive function, spatial cognition, as you can see here, personality change, problems also with language and grammar. So they collected enough evidence to prove that the cerebellum has also a cognitive role besides the motor one. And the current belief is that the cerebellum modulates cognition and emotion in the same way that it modulates motor control. If anatomically the cerebellum is very well characterized, as we saw before, functionally is less known. There was a collaborative study just 5 years ago in which they tried to map the functionality on the cerebellum surface to build a statistical functional atlas of the cerebellum. As you can see here, a lot of functions are reported, not only the motor ones, also cognitive and affective ones. And they achieved this great work, but with incredible fatigue, in the sense that they performed massive fMRI scans. And even if this map is predictive also of individual maps, they noticed that there's a high intersubject variability. And to achieve better functional maps, they needed at least 3 hours of scan per subject. We are talking about functional activation, but how do we measure functional activation? So we know that functional activation, there is an electrical activity. And to work, neurons need glucose and oxygen. And the protein in the blood that is carrier of oxygen is the hemoglobin. So what happens when there is a functional activation is that more oxygen is needed. So with neural activity, there is an increase in oxygenated hemoglobin and an increase in blood flow. So what we would see if we observe the changes in concentration of hemoglobin, we would see that in correspondence of a functional activation, we have an increase in oxygenated hemoglobin and a decrease of hemoglobin which is not linked to oxygen. And then after the task ends, there is a return to baseline. This is called hemodynamic response function, or HRF. So we said that when there is neural activity, there is an increase of oxygenated hemoglobin, but we can go also the other way around, meaning that if we measure the oxygenated hemoglobin increase, we have an indirect measure of neural activity. In neuroimaging, the gold standard for hemodynamic-based functional imaging is the fMRI, the functional magnetic resonance imaging. So, and this allows to measure the changes in oxygenated and deoxygenated hemoglobin because they have different paramagnetic properties. The advantages of fMRI are, first of all, the very high spatial resolution in the order of millimeters. It allows to cover the whole brain. We can acquire both structural data and functional data, and then it's well mastered in terms of hardware and software. But all of this comes with some disadvantages. First of all, the slow acquisition rate. We are in the order of a couple of seconds. Then that is very much sensitive to motion artifacts. And not all the people can undergo an fMRI scan. For example, people with metal implants or also very young children. It's expensive, and as you can see here, the experiments take place in a very constrained environment, in a narrow scanner, which is also noisy and doesn't resemble real-life conditions. In the recent year, there was another technique that is emerging in the field of neuroscience, and this is called functional near-infrared spectroscopy, FNIRS. The simplicity of FNIRS relies in the fact that we can place sensors over the scalp and just inject light into the tissue. So it's an optical technique. The advantages are a better acquisition rate. It's 10 times faster than fMRI. It's less sensitive to motion artifacts. It's inexpensive, always compared to fMRI. It can be portable, and it offers a great recording flexibility. For example, we can see here in the simplest setup, with a source and a detector, which together form a channel, we have this kind of output. So we have the changes in oxygenated and deoxygenated hemoglobin. But if we have a more dense setup like this, we can even perform, as seen in this video, a 3D reconstruction of the signal onto the cerebral cortex. FNIRS has also some disadvantages, like fMRI. For example, the lower spatial resolution. Here we are talking about centimeter. It allows to cover only cortical brain, not deep brain structure. It's more sensitive to extracerebral hemodynamics, and the type of air and skull may influence negatively the signal. But how does FNIRS work? How can we extract these measurements of changes of oxygenated and deoxygenated hemoglobin? So we said we have a source and a detector. We have light injected into the tissues, and what happens? There can be two main interactions. First of all, that a photon travels and at some point is absorbed. Then there is scattering, so a photon travels and is detected back at the detector site. This phenomenon is described by the modified Beer-Lambert law that states that the change in attenuation between the incident light and the detected light, that is called also delta optical density, is proportional to the distance between the source and the detector, another geometrical factor, and the change in absorption in the underlying tissue. We can explicit the change in absorption from this formula. We know also that the change in absorption in the underlying tissue is proportional to the change in concentration of the substances that provokes this change in absorption. And in this case, the two main molecules, or chromophore, are the oxygenated hemoglobin and the deoxygenated hemoglobin. What is very interesting about these two substances is that they have different optical properties in the near-infrared range. We can see, for example, in the lower bound on the near-infrared region, the deoxygenated hemoglobin absorbs much more light than the oxygenated hemoglobin, while after the crossing point, it's the opposite. So what we could do is we could choose strategically two wavelengths, one in this region and one in this other region, to perform two measurements, and then we end up with two equations with two unknowns, which are respectively the delta HBO and the delta HBR. And then we can solve accordingly the system. We talked about cerebellum, functional activation, and FNIRS, but why are we linking these two topics together? So generally in the literature, scalp-based measurements of cerebellar activity were never done before for FNIRS, and were really rarely done also for EEG and mostly in experimental research. And the main reason for this skepticism is, first of all, that the cerebellum is too deep. So there's a larger distance between the sensors that we place on the scalp and the cerebellum. Then that it was believed to be too noisy because it's shielded by the muscles around. This was until 5 years ago, when there was a breakthrough study from Todd and his group in which they achieved successful energy band-based cerebellar electrophysiological recordings. They identified two landmarks below the inion, 5% below and 15% laterally located, in which it is possible to acquire clean cerebellar signal. And so this serves as an inspiration and motivation to acquire also cerebellar FNIRS around the same landmark points. The research rationale is so to develop and validate FNIRS within a new multimodal framework for cerebellar investigation. As first of all, the primary objective is to demonstrate the feasibility of cerebellar FNIRS recording to all these difficulties that we listed previously. And in particular, the first research question is it feasible to acquire it? The second research question is, we acquire something, but how can we be sure that what we acquire comes really from the cerebellum? The second objective is more regarding a multimodal comparison between an integration between the FNIRS and fMRI. Specifically, the first question is how is validated by fMRI? How do they relate spatially and temporally? And what is the impact of the pipeline on the results? And finally, what are also the physiology insights that we can draw from this experiments? To answer this question, we performed 3 studies, which are the contribution of this thesis. So we start with study 1, which consists on a set of preliminary experiments. So first of all, we performed a proof of concept that was successful. And after that, we passed to genuine recording of cerebellar response. So in this presentation, we will focus on experiment 2 and experiment 4. So the experiment 2 of the study 1, we recorded 2 channels. As you can see here, we put source and detector around CB1 and CB2 suggested by the study of Todd. The distance between the source and the detector is 3.5 centimeters. We acquired 3 subjects. So the objective is to detect with a motor task cerebellar activation in 3 subjects that have a full spectrum of lateralization, meaning that they are from fully left-handed to fully right-handed. The protocol consists in the acquisition of a baseline, and then we have a block that is composed of a finger tapping period of 10 seconds and a rest period of 30 seconds. This block is repeated 6 times for the left hand tapping and 6 times for the right hand tapping. So the signals were analyzed accordingly. We removed first the baseline, then we bandpassed the signal of interest, which is between 0.01 and 0.09 Hz, in order to reduce all the physiological oscillations that are not of interest. Then we transformed the optical density into the changes of HDO and HDR according to the modified Pierre-Lambert law that we described before. We performed the time-locked average and then a frequency domain analysis. So these are the results relative to the 3 subjects in which the task period is highlighted in gray. We will focus mainly on the right-handed subjects here. We can see that we have an ipsilateral response, respectively for the left hand tapping and the right hand tapping. What is interesting is also the engagement of the contralateral hemisphere. And this is also confirmed by the frequency domain analysis. So what we should see in the power spectrum of the signal is a peak in correspondence of the stimulation frequency. That is 1 over the duration of the block, task plus rest. For example, here we are showing the left tapping and subject 2 and 3. We observe that we have this peak in the ipsilateral hemisphere, in the left one, and for subject 2, which is right-handed, we have this peak also in the contralateral hemisphere, which is the dominant one. The experiment 4 of study number 1, we performed acquisition of 24 long channels, 20 on the motor and 4 on the cerebellum, and 8 short channels. Short channels are basically channels in which the distance between the source and the detector is smaller and typically smaller than 1 centimeter. And what we acquire is not valuable in dynamics, but only extracerebral signal. The distance between the source and detector is 3 centimeters, and here we acquired 16 subjects. The objective was to assess the reproducibility of cerebellar and also motor FNIRS at the group level. We performed two sub-studies. First, a paced finger tapping task at 1 second, executed by 10 subjects, 8 right-handed and 2 left-handed. And then a not paced finger tapping, executed by 6 subjects, 4 right-handed and 2 left-handed. The protocol is similar to the previous one, except that this time the duration of the rest is randomly variable between 30 and 40 seconds, and the block is repeated 10 times for each end. Besides the classical pipeline that we described before with the pre-processing, for this study we used the General Linear Model Framework, or GLM. So according to the GLM, our measured signal, that can be fNIRS but also fMRI, can be described as a linear weighted sum of some regressors and an error pair. And the solution of this, meaning that we have to find the weight of the regressor, can be obtained by an ordinary least square approach, OLS, accordingly. Another way that is used to obtain the beta solution is an autoregressive iterative least square, AR-ILS. That is basically an iterative form of the OLS in which there is weighting on prefiltered signal. And what is usually done is to test if the beta coefficients are non-zeros, typically with a t-test. The choice of the regressors is arbitrary, and we can include everything that encompasses knowledge about our signal, like experimentally controlled factor or whatever other information we have regarding the signal. But the most common that are used are drift regressor. Here we reported a constant, but usually a qubit is more used. And an HRF model in correspondence of the task. And, the short separation channel that we mentioned before. So to remove the extracerebral component from our measured signal. The most used HRF model is the modified gamma function, convoluted with a boxcar which has the duration of the plot. But in fNIRS, since we have high time resolution, we are interested also in the reconstruction of the signal, and what we can use instead of a simple function is a set of Gaussian basis functions. This is called finite impulse response model, FIR. And what are the advantages of using this is that we avoid to make any assumptions on the HRF shape and latency. As we can see here in the design matrix, we have a set of Gaussians, and each of these has their weight that has to be determined by the GLM. So yes, so each of these Gaussian is a different regressor. So we can define the spacing and the standard deviation of the functions. The results of the experiment 4 for the paced and not paced finger tapping are reported here. We have on the upper panel the left hand tapping, the right hand, and then we have motor cortex and cerebellum for each of the plots. So we can see that for the paced task, we have contralateral activation on the motor cortex. And here we have also ipsilateral one. We have also activation of the right cerebellar hemisphere. For the not paced task, we have activation only for the right hand tapping, and we have contralateral activation in the motor, and then ipsilateral activation in the cerebellum. It is difficult to draw a conclusion on that, as the two sample sizes were different. Here we have 10 people for the paced tapping and 6 people for the not paced. So we decided to join together the results coming from all the left-handed people and the right-handed people. For the left-handed people, we have significant contralateral activation, for the left-handed tapping, and also a minor one for the right-hand tapping. But what we can see already visually is that there is not a marked difference between the two hemispheres, and this is in agreement with the literature. And we have activation of the left cerebellum for the right-hand tapping. For the right-handed people, we have a strong and more extended contralateral activation, and we have activation of the right cerebellum. Sorry. So what we want to highlight, because what we showed before were group level results, is that we detected at the subject level a high variability, as we can see here. These are the HRF from the reconstructed response from 3 subjects, and they differ in polarity and also in latency. So the summary of the study 1 is that we showed the feasibility of acquiring clean cerebellar FES, and that subjects with different level of lateralization may result also in different frequency characteristics of their cerebellar HRF. And we obtained that task-evoked cerebellar activation, both in cerebellum and motor cortex, have high variability across subjects. So we passed at study 2 that consists in the sequential acquisition of FERs and fMRI. Here we acquired 24 long channels and 2 short channels. The setup is very particular, as in this case we didn't make use of a cap, but we glued the sensors directly on the scalp with a clinical adhesive that is called collodion. And the position of the sensors on the scalp are detected through a neuronavigation system, which is a tracking system that makes use of the subject-specific anatomy. So the distance between the sources and the detector here is variable in the range between 1.5 and 4 centimeters. And we acquired 16 subjects. So the objective was to target our region of interest at the cerebellum by designing an optimal montage. The design of this optimal montage makes use of the subject-specific anatomical scan. So we have the scan, we segment it, we pass from volume to surface with an interpolation, we identify on the surface the region of interest that we want to measure with our sensors. Then we simulate how light propagates inside the tissue, and then we solve an optimization problem that ends up with the coordinates of our montage. But what is this optimization problem about? So let's see a simple approach. Let's say that this is our region of interest, and that we want to find an optimal montage for this region by employing 2 sources and 2 detectors. So we have the light sensitivity measurements at each position for the source at a vertex I, and by putting a detector at a vertex J. So the sensitivity is the brightness of this matrix. So what we want to find is the 4 highest points of the matrix, with some constraints, of course, in reality, and specifically this one. But going back to our case, how many sources and detector we should use to measure the cerebellum? We conducted some simulation by changing and gradually increasing the number of sources and detectors. So the results are shown here. We have basically the simplest case that we simulated with 1 source and 2 detectors, and then the case that we choose as optimal with 3 sources and 4 detectors. We can see that here the sensitivity is 5 times higher than the simple one. We simulated also with more detectors, but we found out that sensitivity was kind of saturating at this point. So this is the montage that we choose with 3 sources and 4 detectors for each subject. The shape of the montage was kept for each subject, with this star-like shape. Of course, the montage was either rotated or translated, adapted to the subject anatomy. The objective of this study was to assess the 3D agreement in activations between the sequential fMRIs and fMRI by making use of a personalized montage. So here we performed two tasks, a finger tapping and an auditory task, in which the subjects had to passively listen to speech and noise. The task is as defined before, with the block repeated 10 times for each condition, either left and right or speech and noise. In order to perform a 3D comparison of fMRIs and fMRI, we had to face the problem of image reconstruction. So we have the delta OD, which is optical density, and our fMRIs measurement, our raw data, basically, which is equal to a sensitivity matrix A and multiplied by the delta mu A. The sensitivity matrix is the model that has to be determined, and is basically describe how light propagates inside the nucleus. And the delta mu A is the absorption change we are interested in, and is the value that has to be derived. In order to solve this problem, we adopt a two-step strategy. So we solve first the forward problem, so we obtain this sensitivity matrix by modeling how light propagates inside the human head. And this is typically done, as in our study, with Monte Carlo simulation. And after that, we can solve an inverse problem, which is ill-posed, and extract the absorption change coefficients. We can rewrite our system in this way. Since it is ill-posed, there are different strategies in the literature to solve it. One of the most common used in the fMRIs literature is the minimum norm estimation, MNE, in which basically we make use of a Lagrangian optimization by adding a penalty term here, in which we have the inverse of sigma d, that is the noise covariance, and the inverse of sigma s, that is the model covariance. And we can play with this penalty term, adjusting it, and deciding how depth to go with our reconstruction. Another algorithm which is used in the literature and also in our study is the maximum entropy on the mean, the MEN. So the hypothesis of this algorithm is that there is a prior distributions that describe our system. And the optimal probability distribution, DP, is the one that maximizes the divergence from the prior reference distribution. What does it mean? That in absence of any measurement, the best solution is our prior. But if we have measurements, we want to gain some information from these measurements. We want to maximize the uncertainty that we get from these measurements and increase our knowledge about the system. So basically, we want to maximize these divergences. So the role of the prior is very important. And how do we construct this prior distribution? So talking about the cerebellum, we assume that it's divided into k independent and non-overlapping parcels. And each of these parcels is assigned a prior distribution with an activation flag according to this. So the parcel can, if the parcel is inactive, it can be switched off with a delta direct. Otherwise, if it's active, the prior can, the spatial covariance, sorry, the functional covariance of the prior is drawn from the algorithm we saw before, from the MNE, and it's sigma k. The pipeline we used in this study was very briefly the acquisition of fMRIs, then fMRIs processing. On the other side, we have fMRI and then fMRI processing. We have the acquisition of the anatomical fMRI and the digitalization of the position. And from here, we can solve the forward model with the Monte Carlo simulation. We go and solve the inverse problem, and then we end up with an fMRI schema. On the other side, we perform GLM we end up with an fMRI T-map, and then we make a comparison. More in detail, this is the output of the F-near's source reconstruction. So it's a signal on time for each vertex. So in order to have a single point on time for each vertex, we perform a vertex-wise t-test to test if the signal is significantly different from zero. The mean of the signal is significantly different from zero. And so we end up with a map that we can threshold. On the other side, we have a T-map from the fMRI obtained from the classical GLM pipeline, and we interpolate it from a volume to a surface. So we have two maps. We make spatial comparison by calculating the correlation between the two maps, the dice coefficient, which is a measure of spatial agreement between the two maps, the overlap, and then we calculate the localization error of the center of the cluster for both maps. Then we take the point of the maps that are survive the thresholding, and we average the signal in this point for both F-nears and fMRI, and we calculate the temporal correlation between the two signals. So just to check that there were activations, we calculated the group level results for the fMRI. You can see that for the left tapping, we have activation in the lobule, ipsilateral lobule 456 of the cerebellum, the contralateral primary motor cortex, and then contralateral also thalamus and putamen. For the right condition, besides the activation in lobule 456, we have also an activation in lobule 8, and again we have the primary motor cortex contralateral and the thalamus. For the auditory task, we have, as expected, a strong activation in temporal region, both in the left and the right side, as the task was delivered binarily. And then we have a very small but very strong cluster in the left cerebellum for the speech condition. While for the noise condition, we have activation only in the temporal region. So the results of the spatial and temporal comparison will be presented in the form of a box plot in which we have the red boxes for HBO, the blue boxes for HDR, and then we have the first and the third subgroup referring to the left for speech condition for the auditory protocol, and then the other ones for the right or noise. And for each subgroup, we have the first box, which is MEM, and the second subgroup that is MNE. So in terms of spatial correlation, we see that there is a high variability. So it spans from very high positive value to very high negative values. And but the mean absolute values are above 0.5. The dice coefficient is not very high. There's a slight agreement below 0.3 on average, but the overlap is above the 50%. Regarding the localization error, on average, we are below 30 millimeters, and this is in accordance with the literature. Regarding the temporal correlation, we have again a variability, but the mean absolute values are higher, around 0.6. And we found also significant differences between left and right condition for the HBO cohort with both MNE and MEM algorithms. For the auditory protocol, the results are very similar. We are going to list only the different characteristics. But for example, here we found a significant difference in the spatial correlation for the HBO in the speech condition and the MNE algorithm between the left and the right cerebellum. And this is expected as we have an activation only on the left cerebellum for the auditory task. For the temporal correlation, we have a significant difference between speech and noise for HBO and found with the MEM algorithm. So to summarize this study, we demonstrated the feasibility of designing and using a personalized high-density montage that is tailored to target cerebellar regions. we performed a spatial and temporal comparison of fMRI and fMRIs, and this comparison was surface-based. We have slight agreement with the dice coefficient, but we have moderate spatial and temporal correlation between the two modalities. All the metrics show globally high intersubject variability. So the last step was to perform again a comparison between fMRIs and fMRI, but this time performing a simultaneous acquisition. So we acquired 34 long channels, 20 on the motor and 14 on the cerebellum, and we acquired also 8 short channels distributed over the 4 ROIs. And this time the montage is standardized, and we designed this montage on purpose in order to be sensitive on the cerebellar areas. We acquired 10 subjects and 3 were excluded. The objective of this study was to assess the level of agreement between simultaneous fMRIs and fMRI by using a standard grid montage with a cap. So we performed a sequential finger tapping task, and the protocol is as the one previously described, so with a block repeated 10 times for each condition. So the pipeline consists here in the simultaneous acquisition of fMRIs and fMRI. We acquire also the anatomical MRI, and we digitalize the opted position on the anatomical MRI. We have on one side fMRIs processing and then the GLM pipeline. On the other side, fMRI processing, the GLM. And then we have the co-registration of the two modalities, which is done by calculating the forward model with Monte Carlo simulation. And then we take for each fMRIs channel the maximum point of the cerebellar cortex with maximum sensitivity of the forward model. So we establish a correspondence between the voxel and the channel, and then we perform a spatial and temporal comparison. We wanted to investigate the influence of the GLM pipeline and nuisance regressors on the results. So we investigated 8 pipelines for fMRIs. These 8 pipelines differ mainly for the HRF model, so we used either FIR or the canonical model. Then solver, so we used either the classic OLS or the autoregressive iterative least square that we will call for facility ILS. And then for the presence of an additional regressor, which is taken from the fMRI. So we implemented a dual-based regression in which we use in each of the two modalities a signal coming from the other modality. The pipelines compared for fMRI are varies according to the number of motion regressors. So we start with the standard pipeline that uses 6 regressors, and then we increase by using also the power of these regressors, the derivative, and then the power of the derivative, ending up with a maximum of 36 motion regressors. The pipeline differentiates also by the presence or not of scrubbing, which is volume sensoring based on a threshold for motion artifacts. Then for the presence of additional physiological confounds, which can be the global signal regressor or the signal coming from the cerebrospinal fluid or the white matter. And then here again, similarly to fMRIs, we put a regressor that is coming from fMRIs, which is specifically the PCA of the short channels. So the data analysis is structured in 4 parts. The first part is a task sensitivity analysis. So we perform group level results just to check for activations. Then we perform spatial comparisons, a spatial correlation of beta between subjects. So basically we have the beta map for each subject. We average them, and then we compute the correlation between the average beta map for fMRIs and fMRI. Then we have a temporal correlation analysis. So we take the highest correlated channels within subjects. For each subject, we compute the correlation between each F-Mears channel and the correspondent bold signal. We take for each ROI, we have 4 ROI, we take for each ROI and for each subject the channel with the highest correlation. And then we finally performed an analysis within modality. So we checked if the average beta map at the group level exhibited significant differences across pipeline. So we have pipeline 1, 2, and so on for fMRI, and we checked with statistical tests differences among these pipelines, both for fMRI and for FT. So the results of this of the task sensitivity analysis for fMRI, we are nothing surprising and different from the previous studies in that we have activation in the ipsilateral cerebellum, lobule 456 for both conditions, contralateral primary motor cortex, and also some activation here in the postcentral gyrus. Regarding F-Mears, we have a contralateral activation for both conditions, and we have for the right for both conditions also ipsilateral activation, more extended on the right hand tapping. And then on the cerebellum, we have significant activation both on the ipsilateral and contralateral for the left hand tapping, and as well also for the right hand. In order to check that these activations were genuine, we went to look also to the time-locked averages, so without performing the GLM pipeline. And we can see here the changes in concentration for the motor cortex and the cerebellum. We can see a high synchronization with the task. And if we average the signal without removing the short channel, we can see that we have a marked task evoke change, and that the HRF has a different shape than from the cerebral cortex. The results of the spatial correlation, so before going to quantitative values, we just checked visually the beta maps, and we can see, for example, here in the fMRI, so each plot is the results of a different pipeline, the 8 different pipeline. For example, for the fMRI, we can see that if we go to 36 regressors, we see that there is a change in beta. So here is mainly positive, and here it becomes negative. And this changes also with the F-Mears HDO. When we change the HRF model from the canonical to the FIR, we see that by using the canonical, there is a more better distinction between the two aims here, as one is more positive and one negative, while here they look more or less the same. Going to the quantitative results, so we wanted to find the best matching pipeline in terms of correlation between the two modalities for cerebellar analysis. We found that some correlations were significant, but what is the take-home message from this analysis? Is that the pipeline drastically changed the correlation between the two modalities. So we do not have to choose randomly the pipeline, as the results changes a lot. We complemented this analysis with a temporal correlation. So we can see here for each subject and each ROI, the superposition of the bold with the most correlated HDO, the respective HDR, and then likewise. So we can see that for some subjects in the cerebellar region, bold is perfectly aligned and superposed with HDO. For some other, it is aligned with both HDO and HDR. And here aligned with HDR only. So we can see again a high intersubject variability. And if we look at the correlation for each subject and each ROI, we see that we have, as expected, a significant contralateral correlation on the motor cortex. So for the left end, we have a right motor cortex and vice versa. And we have also ipsilateral one for the right end movement. What is interesting is that we have a significant ipsilateral correlation on the right cerebellum for the HDR chromophore in the ipsilateral movement for the right hand. But here again, we can see that there is a high intersubject variability in terms of cerebellar correlation. But we have again very high positive correlation, very high negative also here and in the upper part. The last part of the analysis within modality, we performed a Kruskal-Wallis one-way ANOVA test in order to check for differences among pipelines, and we found significant differences with post-hoc tests for fMRI and fNIRS HDR chromophore. For fMRI, these differences are mainly due to the addition of motion regressors. So between pipeline which present 6 motion regressors and 9 or 36. And then the other factor that causes this difference is the inclusion or not of the global signal release. For fNIRS, we found that these differences were mainly between pipeline with which were using ILS solver versus the ordinary list. So the summary of this third study is that we have high spatial correlation between simultaneous fNIRS and fMRI recordings by using a generic cap during a motor task. And most importantly, we obtain substantial temporal agreement for all the subjects and conditions between the bulb and the fNIRS HDO and HDR. And interestingly, we found statistically significant differences for pipeline cerebellum analysis, not only in fNIRS, but also in fMRI. So taking everything together, we performed a preliminary studies, and we have to acknowledge that we use different setups and we use a sample size that certainly could increase. But most importantly, our sample size exhibited some variability in terms of handedness and age. So what could be done, at least in this phase, is to reduce the source of variability of the sample size, for example, taking only right-handed subjects. Then the optimal montage is for sure a very elegant and precise technique, but also requires highly technical skills, because if the sensors are not properly glued, this could harm the data quality. And it's a very time-consuming and demanding approach, which is best suited for proof of concept or long-term recording of pathological subjects, for example, epilepsy. Then we performed comparative studies, multiple comparative studies between fNIRS and fMRI, but we have to acknowledge the different conditions in which these studies were performed, because once we performed a sequential acquisition, once we performed a simultaneous acquisition, and we performed once with a personalized montage and once with a standard montage, and most importantly, once we did a 3D analysis without the application of GLM. In the other case, we applied instead different GLM pipelines. And among these pipelines, we need metrics to choose which is the best one. Then we found task evoke change, but also we need additional confound measurements to be sure that these are genuinely neurally related. So if we go back to the initial research questions that we stated, is it feasible to record cerebellar fNIRS? Yes, so our study is a pioneering study, the first study that demonstrates the feasibility to acquire cerebellar fNIRS with all the experiments we performed. And we are sure that we are targeting cerebellum by making use of an optimal montage, as we establish a direct correspondence between the anatomy of the subjects and the scalp measurements that we are doing. And in terms of validation by fMRI, we found a fair spatial correlation between the two techniques that remarkably increase when the acquisition is simultaneous. And we found a moderate to high temporal correlation in all the comparisons. In terms of pipeline, we found that the pipeline definitely matters, and we found statistically significant differences when analyzing the cerebellar ROI for both fMRIs and fMRI. And in terms of physiological insights, we found in fMRI, but most importantly in fMRIs, significant activation that are task evolved. So this work opened a lot of future research direction in multiple directions, both from the clinical side and also on the more methodological side. If we can list some, for example, we can, as we already hinted before, increase the sample size, but also robustify the statistics method of analysis and possibly extend the investigation to clinical populations. It would be very interesting, besides the motor task and the auditor task we performed, to design targeted cognitive experimental protocols that elicit activation in the most external cerebellar lobules, CRUS1 and CRUS2, which are also the most easily accessible by fMRIs. And then, since the vascularization of the brain and of the cerebrum and the cerebellum is very different, it would be interesting to evaluate the influence of the vessel's geometry on the signal by, for example, applying bioentrance. And as we anticipated before, it would be beneficial also to measure additional confounding signals and eventually also other chromophores with additional wavelengths. And most importantly, the most promising future direction is the integration with electroencephalography with EEG, which is already in progress as we acquired in the course of this work EEG with the other two modalities, FMRIs and fMRI. So this work was object of some publications and communications. And here I want to thank all the people that were part of this project, all the people that are here today. It was a very collaborative work. So thank you.
Merci beaucoup. So it's time for the question. So just maybe a remark, we are quite a huge jury, so maybe I ask people from the jury to try to be synthetic in their question. So let's first begin with the two reviewers. So maybe Miss Élise Bagnier, who is ingénieure de recherche at the University of Rennes 1. So can you begin, please?
Yeah. Okay. Can you hear me?
Wait.
Yeah.
Okay.
Yes. Can you hear me?
On ne vous entend pas.
Et moi, je vous entends bien. Mais on a testé tout à l'heure pourtant avec Christophe Grova, je crois qu'on s'entendait. Est-ce que là, c'est mieux?
Moi, je t'entends, il n'y a pas de problème.
Ah. D'accord, merci.
Okay.
Can you hear me now?
Yes.
Yes, good. Thank you very much. Right. Thank you, Professor Descombe. Thank you to the supervisors, and thank you, Giulia, for the invitation to review this work. I very much appreciated reading the manuscript. It was extremely well written. I particularly liked the way you introduced your questions and the highlights at the end of each chapter. I wanted to congratulate you for the challenge that you undertook by investigating cerebellar FNIRs. Well, you tackled two challenges. I mean, once the cerebellum and then FNIRs. I have a few questions. You said we should not be too long. How much time do I have? If you could just...
Okay, 15 minutes.
15 minutes, okay.
20 minutes.
15, okay, thank you very much. So I have, what will I start with? Yeah, you mentioned the high inter-subject variability. Can you elaborate on that? How can you explain this variability? Is this like a physiological difference between subjects? Is it can it also be a measurement error? Maybe you're aware that some subjects have negative responses. Did you have some of those subjects, and can you explain for that?
Yes.
Is it related to the baseline and the use of the baseline?
Okay, so first of all, I want to highlight that the intersubject variability is not something new in the cerebellar study, in that also, for example, the study that I cited at the beginning about the functional atlas, so in fMRI, it's known that subjects exhibit a high intersubject variability. For example, another study demonstrated that they needed the double of the data that respect to cerebral cortex to come up with some functional activation maps for the cerebellum. Regarding some hypotheses, we may speculate that this could depend also on the vascularization of the cerebellum itself, as the cerebellum is very differently vascularized from the brain. In the brain, we have a series of arteries and veins, which are the same for each subject, in the sense that the main arteries and veins and the veins in the brain, we found them in the same position. While for the cerebellum, we have 3 main arteries, and then we have also the big transversal sinus above, which could be also a cause of this hemodynamic variability. But going back to the 3 cerebellar arteries, these are not the same in all the subjects. In fact, in some subjects, we have ramification of these. In some subjects, for example, one of the arteries can be totally absent. So we hypothesize that this could be one of the possibility for this variability. And then, besides these, of course, we still need to be sure that what we measure is truly neuronal related, because for sure it's task evoked, but we have also task evoked systemic change. So we have decided that in some of these, we still might have some physiological contamination, let's say.
Well, I thought that we also had well, trouble having there is a lot of inter-subject variability in the brain as well using FNIRs, right? FNIRs is not really ready for individual use, right? It's we were doing some group studies, so there is some variability in the brain as well. Not like
yes, then I think that also in the variability in the cerebellum is higher with respect to brain, as we have a lower SNR. So probably to reach robust results for the brain, we need maybe less trials, or maybe a different amount of trials, but to reach the same robust conclusion for cerebellum at the group, but also at the individual level, we may need some experimental protocols that are stronger, that are probably longer with multiple trials, and in order to increase also the SNR.
Okay, thank you. You mentioned several times that you had to exclude some subjects. Can you tell us why you had to exclude those, and how did you control for the signal quality to decide on which subjects to exclude?
Yes, so this is a summary.
That was expected.
Of the subject exclusion. So for experiment number 3, so this was experiment we performed at the Hôpital des 15-20. Yeah, sorry. Do you mind if I put okay, thanks. So in this study, we excluded 2 subjects out of 3, which is a lot. For sure, we have to say that it was one of the first experiments we were performing. So the exclusion of these 2 subjects was 1 for fMRI. And especially these subjects refer that he had difficulty in listening to the stimuli inside the scanner because it was very noisy, and he probably missed some trials. For the second subject, it was excluded because some of the channels were noisy. The criteria was visual inspection of the traces, and there was a high systemic components because the cap was a bit small and tight, so the optoids were really pushing into the scalp of the subjects, and the results were really highly contaminated. Even with the GLM, we still saw a lot of confounds. For study number 2, we excluded 2 subjects. So 1 was excluded both for motor and the auditory protocol, and the other one only for the auditory. So 1 for fMRI and 1 both for FNIRS and fMRI. So specifically, 1 subject was excluded because it was the very first experiment we performed, and the field of view in the MRI software was not correctly placed, and we had an exclusion of the top part of the motor cortex. And the other subject of fMRI simply did not complete the auditory task. For FNIRS, it was again the first subject we acquired, and especially on one hemisphere, we had a very high gluing error, meaning that the sensors were not glued in the correct theoretical position that we found with the optimal montage. And I take the occasion to say that, in fact, it's very difficult to glue the sensors, and you need to be a skilled technician, and you need a training phase. So it's not surprising that the first one was not very good. For the third study, we excluded 3 subjects. So 2 for FNIRS, because more than 80% of channel were pruned. So again, we used a mixture of visual inspection and then scalp coupling index. And the other one was excluded from fMRI because he did not complete the task.
Mm-hmm. Okay. Did you use some kind of indicators like scalp coupling index, or was it only a visual exclusion on the FNIRS data that when you did your selection?
No, it was a scalp coupling index for this study 2 and study 3. For the previous studies, since it was mostly exploratory, we did visual inspection.
Okay. Thank you. You've been using a variety of systems. I'm not going to ask you what is your preferred system, but what are the main differences between those systems in terms of usability and like and signal quality? Did you see a lot of differences between them?
Okay, yes, there are for sure a lot of system, and we can say the preferred one is from NearX both system. And yeah, the difference is, so main difference, first of all, like this one is a device that now is mostly obsolete. I'm not even sure it's still produced by the company, and it's recommended to measure muscle oxygenation. We used it simply because it was available at the university facilities, and so we decided to start the experiment with this. Then we had the opportunity to use the NearSport 2, and this is a very good device in that it can be portable. So if someone is interested to perform protocols also in situations in which movement is required, or also for hyperscanning and so on, It's a very flexible device. And most importantly, you can choose which kind of montage you want to apply. You can go from a simple montage to a very dense montage. So you are allowed to do channel space analysis or also perform the reconstructions. And you can also customize the pressure of the optoids onto the scalp because there is a spring-loaded system. The BrainSight device is really good if you want to perform, as we did, neuronavigation. So if you have, because you have, besides the system, also a neuronavigation tracking software. So basically you insert the anatomical MRI scan of the subject, and then with a sort of pen, moving this pen, let's say, on the subject head, you can navigate simultaneously on the screen and seeing the anatomy that you have underneath the sensor that you are moving. So it allows to place the sensors basically wherever you want on the scalp. You can reglue the sensors, but we have to say that you can also use their sensors by using a cup. So in this sense, is also a flexible system. And there is a part in which you have to calibrate the intensity of the sources and the intensity of the sources. So this requires, yes, a calibration from the user. And regarding the last device, I would say that the main advantage is that it's MRI compatible. So you can perform simultaneous acquisition. And again, you can go from a sparse montage to a more dense one. So more or less, that's a panoramic.
Okay, thank you. I had another question relating to the temporal correlation that the results that you showed. Did you account for a lag between the bold signal and the HBR, for example, or was it
No, I didn't account for the lag. I computed only the correlation. I didn't compute the cross correlation.
Okay, all right. Next question, if I may. You mentioned the future works and the idea of working with EEG. What would be your idea there to combine EEG, FNIS, and MRI, the three all together, or maybe only two of them, and why so? Which kind of?
I think at the beginning it could be interesting. I mean, the integration of EEG and fMRI is already feasible. It's nothing new. And it's possible. We performed the simultaneous setup, and there was some moment in which we felt particularly ambitious, and we thought maybe we can insert also some electrodes into the scanner. But it's possible for sure. It's intricate to perform both a high density FNIRS and a high density EEG, but it's possible. At the beginning, it would be nice to perform preliminary experiments with both the three modalities, but in the long term, it would be for sure more interesting to perform only EEG and FNIRS also, as we said, with other cognitive protocols outside the fMRI environment. The information is very valuable from EEG because we have another kind of, it's another technique. We are measuring electrophysiological recordings. Hemodynamics is measured either with magnetic or optical techniques. So we have another physical principle, and especially we have another temporal scale. And it's, let's say, a closer look at the neural activity because hemodynamics is always an indirect measure. So that's why we think that it would be very useful to perform integration with EEG. And if I may say, so we started to record the EEG with the personalized montage. So in this phase, we acquired only two electrodes, one on the left and one on the right hemisphere. And we didn't analyze it for two reasons. First, for timing reasons, we decided to focus on the FNIRS and fMRI. And then the quality of this EEG for more than half of the subjects, always due to the let's say the hurdles of gluing and so on, was not very good. But in the final part of the thesis, we performed also acquisition of EEG only, and we acquired 6 electrodes on the cerebellum. It's not fully analyzed yet. We just had a look at it, but the signal is very good, and we saw some changes in energy, in the frequency. So in alignment also with thought, for example, already when we open and close the eyes, we see that the alpha power is different from the occipital region because we recorded also the occipital region. So that is more or less the interest in acquiring EEG.
And in terms of future applications, do you already have some applications in mind? You quickly mentioned epilepsy. Is that some, well, do you think there's a interest there for FNIS recording for diagnosis or for follow-up, or what is the rationale there?
Yeah, for sure, it's interesting to measure the FNIRS in epilepsy. For example, one project that is already going on with our collaboration in Concordia, they're measuring FNIRS during sleep in epilepsy, because they hypothesized and they also confirmed that the hemodynamic response for epileptic people in correspondence of the epileptogenic focus is different in the different stages of sleep. So this is a very specific application, but for sure, epileptic people have different hemodynamic properties than the control one. But also there are other pathologies that are related to the cerebellum, for example, also ADHD or multiple sclerosis. It's still, let's say, something relatively new in the literature, like the role of the cerebellum in cognition was established 20 years ago, and now, let's say 10 years ago, they started to establish also a role in pathophysiology, but for sure it could be interesting to measure other kinds of clinical population.
With fMRI, FNIRS, EEG, all three of them? Oh, yeah, you think?
Yes, let's say preliminary, yes, and probably then go out of the scanner.
Okay. Just checking if I have a very urgent questions. Other than that, I think for the sake of time, I will just congratulate you again for the very clear presentation and the answers to my questions. Some of them were expected, I could see from the backup slides. Thank you very much again, and congratulations.
Thank you a lot. I don't know if you want to maybe switch to the microphone. Okay, so I can rephrase the question to the jury. Okay. And also on Cerebellum, because you know your background, it's kind of giving you that point to us now in 2016, and you can also come back and then see your so that's beautiful. And also in the experimental contributions, it's really for every team inside, why you find it how to think about and that's a really of that I was one of the people who were participating in Boston about the Cerebellum and academy story. So really excited how people thought the first time they're challenging it was to be that company in the experiment. So that's a beautiful time and that's many questions are used to do by me. So probably one understanding of maybe I get we didn't say the word ethnic modification that we recorded on the third one. Yes. Yes. So I will show again a picture of the montage. So we are talking about why the motor data that we acquired with the the personalized montage on the motor cortex were excluded. So I would like to, okay, I will do like this because it's easier. Okay, so let's start to say that on the motor cortex, we decided to put data only for control. We did not really, yeah, okay. So here we see the setup. We see here that we have a dense montage on the cerebellum, but we have on the motor cortex only one channel. We decided to place one source and one detector as control. So just to see the typical contralateral activation on the motor cortex. The reason why the signal was bad in most of the subjects is twofold. I'll be completely honest. The first one is because, as you can see here, when there are multiple sensors and they are glued all together, it's like they are holding in place like a patch. When there's only one sensor that is glued or two, and they are maybe not even touching each other, they kind of twist if they're not correctly glued. So this is the first reason. The more sensor you have, the better they will be held in place. The second reason is because this process of gluing took, first of all, it was done by two people, me and another colleague, technician. And this was the last sensors that we glued. And there is fatigue during gluing process, we have to say it. So the first sensors you glue are glued very well, and then the accuracy decreases. These were the last two, so partly due to the less number, partly due to the fatigue, the result, the signal was most of the time pruned. But in these subjects in which it was well glued, the hemodynamic response was classical. So increase of HPO and decrease of HPR. There was nothing particular. Just we didn't report it because this channel survived, if I'm not wrong, in 3 or 4 subjects out of 15. No, yeah, we did not really analyze it. So we did not about the of the paradigm, because there was another something that I did not really think about mentioned that instead of proposing central planning or lower settings, but there is a difference people would have to sit on forward models and that we would be able to measure. Okay, so we are talking about formulating, designing the possibility of designing cognitive experimental protocols for the cerebellum involving a sort of prediction like it happened also in the forward model. Yes, I think this is totally possible and interesting because what's emerged in the last year of research is that the mechanism of the cerebellum for controlling motor and cognitive functions are the same. And this is a reflection, this is just a general comment, but it's reflected also in the site architectural structure. It's so regular, and in its how to say, its mechanism for controlling both motor and cognitive functions. So it doesn't matter really the function, it's more a matter of control. And going back to the forward model, so the forward model states basically that we have a prediction and then an error processing. So the cerebellum is a controller, it fine-tunes. So I cannot think right now of a specific cognitive protocol, but I would play about expectation. For example, I would design something in which there is a stimulus with an expected response, and then you can kind of fool the participant so that you can see the major activation in the controller, because there is an adjustment due to the error that you actually have between the prediction and then the sensory feedback. So this is something I would think about.
Another question. You said that you should start defining the for deciding the guidelines and what
So regarding the metrics to find the best pipelines, in the literature, there are already some metrics, but only for resting state fMRI. So these metrics, it's easier if you want in that case, because they look, for example, at nodes that stays or disappears with the different pipelines. And regarding already the influence of pipeline in task evoked, there's not really literature about this. In affineurs, not really. They tried only, for example, with simulated data, and they changed the solver between OLS and the ILS, and they checked, for example, for the sensitivity and specificity, but they did that with simulated data. With real data, I found in the literature a recent study of 1 year ago in fMRI, in which they tried the different pipeline, and they were simply looking at the zeta map, the zeta values of the statistical map. And then they were drawing some conclusion based on that by making statistical tests and so on. I think this is a bit, probably is the first approach that you can use. It's the most straightforward, probably not the more robust draw conclusion. So regarding the metrics, this is something that can be object of research. Thank you very much.
Thank you, Mr. President. Thank you very much for this presentation. I discovered Cerebellum. You mentioned this information, and I want to come back to that. I think that I'm going to say, already for the control channel.
Yes.
Of course, I have a question about this. And I guess you are not and can we go back why why
Okay, so we decided to compare also mini with MEM because we saw from previous results from our group in Concordia that MEM was very precise in the localization of the cluster. And since cerebellum is smaller than brain, we said, why not? Let's see if maybe we can achieve a better precision with this method. So this was mainly the main reason. And then yet we have also a term of comparison with MNE. So, but the main reason was probably the spatial precision of the algorithm. Okay, so in the whole cerebellum hemisphere, so also the part that is not accessible, let's say, by engineers, we have 1,000 vertex. The part that is, let's say, targeted by the montage, we are around 300 vertex, 400, and it depends on the subject. So the parcels, we performed, as is already implemented on Nearstorm, data-driven parcellization. So with the MSP score, multivariate, I don't remember the. Yes, so sorry, I didn't remember the acronym. So basically, there is an attribution. So there is an attribution of this score, let's say this probability of activation to each vertex of our field of view. So and then we take the constraint we use is to define a number of clusters which is equal to the number of channels used in the montage. Then from here, what happens is that we take the local maximum of the MSP, and then with a region growing algorithm, the parcels are created. This is variable in the sense that normally, so here we have 12 channels per hemisphere, but sometimes some are pruned. So I think this is also one of the reasons why the results were not sometimes optimal and are also highly variable. Because in some hemispheres, in some subjects, we had more pruning than in other ones. And in some subjects, so they were not excluded completely by the study since we had already few subjects, very difficult protocol and so on. So we said we were very relaxed with the exclusion criteria. We kept almost everything that we acquired. But in some montages, due to the pruning, sometimes we have very few overlaps of the montage. And we know that overlaps is basically what enhances the accuracy of our reconstruction. So.
Functional mapping that I know that there is a variant between
So regarding the possibility of integrating the King's parcellization, so the King's functional atlas for parcellization of within the MEM framework, yes, I think definitely this is a solution and it's probably the way to go. Because we noticed that between subjects, there was variability in the parcellization. So I think for sure, the data-driven parcellization with the MSP is a good method. But in this case, when we have something that is smaller than the brain, and if we use also the constraint that the parser depends on the number of channel, we end up maybe in some subject with a very big parser in the center and then other smaller parsers around. So to be more fair, yes, we should use something that has, let's say it's a functional prior, let's say it's a functional, not prior in the sense of, let's say, a functional information that we have about the King's Atlas. And for sure, at the moment, this is the only task-evoked atlas we have, with a very extensive number of tasks. There are also other atlases, but they are mainly focused either only motor or cognitive. They employ, let's say, around 40 kinds of tasks. So it's probably the richest functional atlas to learn about the cerebellum. So I think it would, like using another kind of parcellization would definitely benefit the source reconstruction.
I was asking about that also when you already manifest there's some related about that.
Yeah, I think that is, again, it's a simple approach, but it's not it's not something untouchable or holy. Like we can for sure work on that. And for example, other studies, they use the Spearman correlation. It's not about delay, but it's another kind of relationship. So for sure, yeah, I think it would be interesting because we know also that we have already some delay between HPO and HBR. So it may benefit also to use some kind of cross correlation or something similar to take this delay into account.
And you have some delay between HPO and HBR. And then you also find the right on the hand and the left hand.
Sorry, which experiment? Okay.
That's the response. Right hand side of your,
Maybe I can plot it. So wait, I have to share I have to stop sharing the Zoom. Okay. Okay, so it's experiment number 3. So it's lagging. Okay, yes. Yeah, so regarding the delay in the experiment number 3, to be honest, we didn't really focus on that because since it was results from a single subject, it was difficult to sorry, I got distracted because I saw the chat on Zoom. So it was difficult to assess whether this was due to something that was, let's say, neurally related or more to systemic compounds, because we know that when we do group level results, yes, because so we had 2 were excluded. So let's say that in this study, we mainly investigated the pipeline. So how to use GLM, what was the benefit of regression or the HRF, the GLM solver. but we did not focus on that. And to be honest, I would not know how to interpret this on a single subject.
You can go back to the slide. So, at line 59, we come back to the relay left from the net into the process and the cerebellum. The function
Okay. Okay. Mm-hmm. Yes. Yes. So to interpret this delay between the HRF shape between the motor cortex and the cerebellum, one explanation could come from the study of Todd itself, from the EEG study. So in this study, they found something very particular, that the cerebellum operates in a frequency band which is much higher than the brain. So, I mean, not only in that band, but there is a modification of frequency content in the band that is called ultra gamma. So we arrive also until like more than 200 hertz. So it means that it works faster, if you want. This could be one explanation for this earlier response. And yes, that could be one explanation.
After all, you want to look around and see the cerebellum for
Yeah. Yes. Okay. So basically, here we have the time course for each vertex. We perform. No, you don't see my pointer. Sorry. Okay. No, you see. Okay, so the point is that we have, for the fMRI, we have already a T map. For fNIRS, we did not perform GLM, we perform source reconstruction. So we have a time course reconstruction for each vertex. In order to compare one object with one object and not one object with a time course, we performed a t-test for the time course of each vertex. And so we end up with a t-value and a t-value. Then we thresholded these t-maps, both. We did it with both chromophores separately. And so we threshold these two maps, both for fNIRS and fMRI. So we have this thresholded map. Then we take only the point, the vertex of the thresholded map. We take the time course of this vertex and we average them. So basically, we average the time course of the activated vertex. Yes.
I think it is very interesting the fact that it helps more agreement in the environment. So there is some hope, I think, to make more agreement.
Yes. Yeah, because here, of course, if already the two maps are not the exhibit cluster in two different places, we are already comparing two signals that comes from distinct places. So, I mean, there are this different strategy to do that. Like we decided this, but for sure there are other possibilities that are also reported in the literature.
I was fascinated by this functional assessment in Cerebellum because for each transfer that we had also a functional attribute.
Yes.
It was not really,
Yes.
so do you think that we have to try to figure out that these are and we'll keep that. Can you think that in this sort of technique, because we personalize like the third, you think it could also available related to attributes that is probably more as well. This concept is really related to a particular type. You think it could be interesting to not to bias, but it will also some information about what kind of activity at this time it started related to some activity that are something affecting activity. There is a lot of things that I can find it.
Okay, so regarding the possibility of introducing, let's say, also a sort of flag. Yes. Yeah. A sort of label, yes, to the functional atlas regarding the task. Yeah, I think it is a good idea, but regarding specifically the King Atlas, we have to be cautious in that the three labels that they put, they basically choose the main three tasks that elicited area activation, in that area. But actually activation was elicited also by other tasks. So they made some probabilistic computation and attributed to each parcel three main, let's say, types of activation derived from their task. But so we have to take into account that these are only three of the many, and also that they performed, even if many tasks, but only determinate tasks. So in real life, there are also other kind of tasks. So I think, yeah, it's a good idea, but we have to be cautious on how to attribute this. Yes, yes. If we are too strict, we may miss something. Yes.
So my last question: how do you think that what is the done
So with EG, we started with two, one for each hemisphere, like the Todd did with in the very first study. But now after Todd, there are a couple of other groups, let's say like 3 or 4 groups in the scientific community that is trying this. So for the last recording that we did, we acquired 6 electrodes. So we put 2 in the midline, and then 2 on one hemisphere and 2 on the other hemisphere on 2 lines. And I think the first objective that we could aim at regarding the EG, rather than already going for source localization and something more complex, to replicate what Todd did. So an energy-based analysis. So to do some kind of block design task, to record the response in these long blocks, and then eventually see the variation of activity of the various EEG rhythms and that perform a time frequency analysis.
Thank you very much.
Thanks.
So now it's Professor Moon from the technical. Thank you. So lasers work, they investigate the cerebellum with needles. None of those very popular, but very important the first question I have for this, all the time for your dissertation, LinkedIn and modeling for students, particularly for the introductory chapter. So how careful you describing the theory of the imagination, and so on. One of your operational choices is funded, and this is with the emphasis on enhancement, because these left-handed person with such a field is so hard to find the left-hander, and almost so it added a lot of variability to their study, stop the samples in the middle or even more less enough. What was the motivation for how this the randomness entered your investigation?
Yeah, so the reason for introducing endedness at the beginning stems from experiment number 2, in which we had initially, so with the first device we used, we had some difficulties in recruiting participants and in achieving good quality signal. Then we let's say carefully selected the subjects that could give in which the FNIRS acquisition was possible with this device. And we had 3 volunteers, and one of them was left-handed. So we were encouraged from this preliminary experiment because we saw that there was some difference between the right-handed and the left-handed subjects. Then we saw also that in the literature, in FNIRS, but also in fMRI, the study of left-handed is very rare. They mostly choose a group of purely right-handed subjects. So at the beginning, we wanted to check for these differences, lateralization for this, to have an element of novelty. And the reality is that when we went to recruit for participants, also in study number 2 in Montreal and study number 3, we had a lot of difficulty in recruiting. So at some point, we basically accepted all the people that were, how to say, eligible to do the experiments because the time frame was very short. And so we thought to add them almost as a case study, like acknowledging the variability that they were introducing in the group level. But I totally agree with you. And if I could do the thesis again from the start, I would reduce all the sources of variability from the data set. I would choose either only right-handed or only left-handed. Also in terms of age, we were a bit large to favor in recruiting of participants. And we started we choose to recruit people from 18 to 45 years old. But also in the simultaneous study, the last one, in study 3, we had 2 subjects that were 44 and 45. And we noticed also from the anatomical MRI that there were some features that were different in terms of...
Sort of fast is like to do the cerebellum. So of course, the technique and the plan to do some level of activation in the cerebellum. But the aim was to provide a piece of principle that is works. So I have to research a task with good physics. You do it with one bringing the activation level of the cerebellum to the need. And we have to do the field is requirement.
Yeah, so the motor task is for sure one of the more robust to elicit cerebellar activation. And the auditory task, it's true, it produces an activation which is very strong but also very localized. So probably if we want to observe something on the near fMRI, it is okay. But on the nearest level, if you want to observe something that is extended and more robust, probably it's not the best task ever. We performed also another task, but it's not yet analyzed. It's completely different because it's not block design, it's event related, in which it's an auditory sound discrimination, because we saw in the literature that they did fMRI and PET studies in which they had very strong activations in the cerebellum when the subjects had to detect differences either in frequency, in pitch, or in interval sounds. But yeah, I agree that we may find some different protocols, especially because even if the motor one is very robust and we have an activation at the cerebellar level, both with FNIRS and fMRI, we see that the activation in fMRI is more towards the lobule 6 and lobule 8. There is also something in CRUISE 1 and CRUISE 2 that emerges more at the individual level rather than at the group level. And we know also that CRUISE 1 and CRUISE 2 are the lobules that are most easily accessible by FNIRS, because we have to be honest, with FNIRS, we cannot access the whole cerebellum. We cannot acquire the vermis. We cannot acquire the intermediate level. We are more on the lateral one. So, of course, for this kind of preliminary and exploratory studies, it would be better to start with some kind of task that elicits activation in that areas.
Next question is related to there are some studies in that the effect of gravity on signal intensity, at least with these tends to be differences, and then depending on the type of disease or are environment without gravity, so then you may see differences.
Yes.
Is there perhaps a mechanical way to express the cerebellum more towards where the sensors are?
Yes, it is already the case actually in the simultaneous experiments, because the subject is laying on the cerebellum. But this is this might be a problem actually, because if you push too much the sensors, you have physiological confounds. You might have physiological confounds. So it's good to, for sure, ensure a good coupling between the sensors and the cerebellum. But yes, we could control more these conditions. For example, they showed also in other studies that it would be beneficial to measure the partial pressure of CO2 or the arterial pressure, because these are elements of confounds that could negatively impact or simply impact the FNIRS signal. So yeah, I agree totally that the position of the subjects is important, because when you go in the fMRI, even if you have optos on the motor, it doesn't really matter because this part of the head is not in contact. But here we are totally laying on the cushion, so there is an influence. If on one side it helps, on the other side we have to be cautious that there's not too much pressure.
How you calculated the correlation to be defined for the years and the fMRI are not at the same length. So this is interpolate, and these are the basis of how many disease regions you in time terms you have at the end.
Yes. So for the calculation of the correlation between FNIRS and fMRI, since they have different sampling frequency, we decided to up-sample the bolt by performing linear interpolation. And we choose the epoch of the time-locked signal. So here, for example, we can see from -5 to 35 seconds. And the sampling frequency of the NIRS if I'm not wrong, in these studies, so was, I think, in this 10. Sorry, the computer is a bit slow, but here should be 10. Yes, exactly. So in this one, we had 400 points.
I make a last point because what we knew from the pipeline analysis, so there's also less component. But in the years, research, we are prepared, so we have the practice model, so it's like better ability properties, right? But we didn't apply the same to the FMRI data. Even if you many of the correlations that we have.
So it's this is a very good comment, because when we compare the pipeline of FNIRS and fMRI in the last studies, we didn't choose the pipeline by varying the same parameters. Besides this, for example, also the HRF model. In the FNIRS, choose FIR and canonical. In the fMRI, we kept only the canonical model. But there's actually the option also already implemented to use the FIR. It's just that maybe it's not that interesting, as the temporal resolution is not the same as in FNIRS. But for sure it's possible. And in terms of GLM solver, as you said, also we use the autoregressive and OLS solver for FNIRS. For fMRI, we used only the OLS solver, the classical solver. So this is for sure a future research direction that we could adopt, like to make a more thorough comparison between the two modalities by, let's say, expanding the range of pipeline that we could use. Because there are serial correlation by physiology. Let's say that in FNIRS, this is highlighted because the sampling frequency is higher. So we have the noise that is even more correlated. Let's say the serial correlation are more important. But this, of course, this phenomenon is present also in fMRI. You're welcome. Thank you for the question, of course. Christophe is muted.
We don't hear anything. Is it my turn? I don't. We really don't hear anything from the committee, from the jury. So
sorry for that. I'm really sorry. Yeah, I saw. Thanks.
Thanks, Jérôme, for at least we have a big follow-up. Okay, thank you. So first, I guess as the others, I would like to congratulate you, Giulia, for the very nice work and very nice, elegant presentation you did today. And also because I know that how difficult were those experiments, especially those we see now on the screen with the collodion. And you did very well, not only in data acquisition that you did in our, while you were in Montreal, but then in analysis. And this was particularly, it's a great job. So I do have several questions. So I try to follow a bit the comment of Jean-Marc on MEM. I will try to continue on a little bit on that, on possible suggestions, and then I'll have a question. So you mentioned, I know we've been saying that it is a kind of idea that the number of parcels in MEM is roughly the number of sensors. But this is by far not a strict rule. And we have shown in EEG and MEG then the method remains stable, but it's full brain, high density EEG, MEG, but the method remains stable when we had less large parcels or many more small parcels. In this context, I would be really quite interested, and perhaps we should also evaluate on simulation, because you don't have that many sensors, it's local, I would give more flexibility to the model. So probably in increasing slightly the spatial mesh, like 300 point is not that much. You can increase locally. And increasing also like changing the neighborhood order to have more parcels. Because having like, I don't know if it's 12 or 15 parcels, there will be relatively large compared, and as you say, this might really drive a lot your results. So I think you will have more flexibility and interest. So it could be, as Jean-Marc said, based on an atlas, or it could be from the data as well, but the algorithm should be quite stable with more parcels. So I think that could be an idea to perhaps investigate in the future. But as the main questions behind that, because as you said, you are showing really average result. And so you pointed out that you are showing a spatial average for MNE, for MEM, which might be a bit biased because it can change from one. And I'd like to know if you look into your data, because as you're aware, inverse problems are highly dependent on the SNR of your data. And we discussed that there is a great variability in NIRS. So I assume that some of your subjects might have good SNR data, some lower. And I'd like to see, of course, if you are localizing almost nothing, like very bad average data, both MNE and MEM, I'm not expecting them to provide any result. And I was curious to know whether when you consider perhaps there were only few, I don't know, cases which decent SNR data, so decent average on the scalp, if you investigate the resulting spatial map, and what were your thinking, your thought about those spatial maps when we are localizing high SNR data, which means we are in good conditions.
Yes, for example, so we can, the example I shown here, this was one of the best subjects we had. You can see, in fact, that the reconstruction also, this is HBO, and the reconstruction is very nice, and also the map is, the time-locked average is very similar in terms of in terms of time course to between FNIRS and fMRI. We have different properties with undershoot, after, and so on, but regarding the activation delay is very similar. Also, the spatial comparison is similar. In fMRI, we have this part that with FNIRS we cannot access. But yes, I totally agree with you that these, like in subjects in which the SNR was better, in which we had almost all the channel that survived the channel pruning, the results were consistently different. So we could also in the future perform either a case study analysis on those subjects and see them individually, and rather than go for group level results, because it is a matter of fact that the quality of data changes changes a lot across subjects. And in fact, like I reported, maybe I can do, I reported a table of statistics of data pruning.
Mm-hmm.
So
Okay.
for each subject, we can see that in some subjects, like only the 16%, or yes, this one were the best one. But in some others, like this one, we had almost one hemisphere that's in which there were no overlaps, and yeah, so we can see that also this other one was not very nice. So we can see here that the number of channels are pruned are much more than with the standard grid montage, in which almost no channels were pruned. And it varies across subjects for sure. In future analysis, also for publications and so on, we could try to exclude a couple of subjects. I wouldn't go less than 10, because otherwise it really becomes a case study. But we could be, if we have a more extended sample size, we could be a bit more conservative on the inclusion and exclusion criteria.
No, these are right points, and it comes to my next questions where we talk about variability. And of course, you have inter-subject variability. Your two study, whether it's simultaneous or non-simultaneous, it's very nice. What we know also from NIRS, and it looks that it is even more the case for cerebellum, is that we have also sensitivity that differ from subject to subject. Some are very good NIRS signal, some for different reasons have bad FNIRS signal, and we have to cope that. You did not talk that much about how you analyze the data in terms of motion. So because of course motion is has huge impact, and the problem with NIRS when we compared for EEG and MEG, where data are more faster, so we can make a lot of average to get a decent SNR data. Usually we have block design with few blocks. So yes, that's okay for fMRI with GLM, but for averaging, we're averaging very few. And if you have two blocks with motion artifact, that's going to create a lot of problems. And so I wanted you to comment a bit about that and how this could impact your data.
Yes, so first of all, I would like to make a comment on the motion correction algorithm. So that it's here, sorry, it's not that big for the audience here, but so we chose the TDDR algorithm, and this is as you can see here, a trace which is not that has a motion artifact here, another one here, and so we decided to correct for motion by qualitatively comparing, visually inspecting all the motion artifacts correction algorithm that we have in the literature. We chose this one, and we checked from subject to subject the trials before averaging and then solving the inverse problem, and we ended up with discarding almost no trials, probably one or two trials in the entire data set, because given that we had gluing and that the subject's head was stabilized on head cushion, there was very limited possibility of movement. But I agree that for future experiments, we definitely need a higher number of trials. And one idea that we could do, for example, as it was done already in the previous studies in neural laboratories, for example, to focus on only one condition. So instead of doing left-hand tapping and right-hand tapping, we could start to try only right-hand tapping and already double the trials by maintaining the same protocol length.
Yeah.
So, but yeah, we definitely need that. And then to strengthen this result, we could also try more robust methods, like, for example, bootstrapping, to artificially increase the number of trials.
That was it's a very good point. That's where I wanted to go with the study we did with Zheng Shen on finger tapping. So indeed, if you bootstrap among those few trials, even it's very good that at the end with the motion correction, you did not have to discard many trials, but at least you could generate slightly different average that are in theory coming from the same distribution. So in terms of statistics, to make source reconstruction, this is way better, and probably to make statistics for your comparison, instead of relying on one average and one, like you did your vertex-wise T-map for MNE or MEM, you could think of something a little bit more robust and something that looks like a true statistical map for your comparison, and that might indeed make the difference between the approach.
Yes, I saw also in a recent study in which they used the NearStorm, and they also perform a surface-based analysis, that they did not reconstruct, they did not solve the inverse problem by using the averaged epochs, but they reconstruct the whole signal. so that they could perform the GLM and then end up with a team up that is obtained in a similar way, and not as we had that one has GLM pipeline behind and one not. So this could be also another direction.
We did that with Edouard for some finger tapping data, but yeah, that's is also a good point. And yeah, so I will not spend too much time. I think you answered very well. Perhaps the last more generic questions is about the motivations behind that work. So it's very good that you show the feasibility of making measurements of the cerebellum. You put fancy experiments together. But at the end of the day, can you explain us why? So what can you do? Let's say you have your perfect protocol for FNIRS, you have excellent results. What is the end goal that you can do with NIRS that you could not do with fMRI? For instance, in the context of cerebellum, what kind of experiment you would like to do that are impossible to do in fMRI?
Yeah, I think probably the major advantage is to perform experiments outside the scanner in real life conditions, because once we acknowledge the fact that the cerebellum has also a cognitive and especially an affective role, as we saw also in the production of the patients of Dr. Schmamann, these are for sure things that cannot be tested inside an fMRI environment. So it is sure that, of course, FNIRS doesn't have the same spatial resolution, and we cannot acquire all the cerebellum, but it could be useful to start some kind of monitoring, first of control and then of pathological subjects in real life situations. Now in FNIRS, there is this trend, for example, of hyperscanning. This maybe is a bit far, but already in the standard montage that one has, in the high density, you could simply add a line of optoids below the inion. So this would be already a simple and fast, let's say, advancement in the common setup that we are, because the cerebellum has always been discarded, basically, even in the high density montage, when we have 64 or 128 sensors or electrodes, it doesn't cost much to add some optoids below.
And on that, there's huge literature studying gait and balance with NIRS, but mainly on prefrontal regions. So showing cerebellum activity there would be quite interesting.
Yes.
Okay, so I will stop for now. I think thanks again, and congratulations for the great work, Giulia.
Thank you very much.
I think that I will consider that good aspect
Yeah, I think for sure cognitive factors could be a source of this variability. And we know really few about that. Like we know, for example, that the brain, like when we talk about cognitive function, are usually located unilaterally. For cerebellum, we start now to investigate this field with the King's Atlas, for example. So, yeah, it could be, for example, like this could be one source of variability for sure. From the motor protocol, it's difficult to assess that, as this was purely motor protocol. We did not have much cognitive involvement. Maybe in the sequential finger tapping task, because we performed like at the beginning it was just tapping, then it was like sequential finger to double position, and also in the paced finger tapping task, because when there is a pacing stimulus, we have to encode time information into a movement. So there is some kind of cognitive involvement. It's not immediate, it's not evident, but this could also be a source of this variability. For sure, like more experiments with other protocols would benefit this hypothesis. So, yeah, regarding this, for example, there is a study in the literature, an fMRI study, that is relatively old, if you want, it's from 1999, in which there is a theory that I support, in which, because we observe not only ipsilateral activity in the cerebellum, but we observe very often also a contralateral one, especially at individual subject level. So in this theory, they hypothesize that it's not related to pure motor control, but it's, for example, the left hemisphere that controls the precision and the specificity of the movement. And in fact, I put it here in the slide. So the activation. Sorry, it's very ugly. You just need a screenshot of this. But they observed basically that the activation on the cerebellar hemisphere was lateral, and on the right one was more intermediate. So they hypothesized also in accordance with previous studies also made on animals, then one, rather than the control of a part, one would be more in control of precision and specificity, and the other one more in control of oscillations, of automatic movements. And so this could explain also why they are mostly bilaterally activation also in our class. Yeah, I think that if some subjects, let's say, are problematic, they are probably problematic to undergo also fMRI scan, because we know that we are, for example, already to constrain them still, and then suppose subjects would have neurological disorders or complications in which they wouldn't feel very at ease in the scanner with the noise, with the also maybe if someone is claustrophobic and so on. So it would be interesting at least to translate these kind of experiments into a more naturalistic environment. And as I anticipated before, if one can acquire, let's say with a cap, fMRIs on the, let's say on the occipital temporal region, which is commonly done, adding some electrodes below wouldn't be much difficult. Of course, the accuracy of fMRI is higher and so on, but it could be a start to investigate clinical populations. Then it always depends on which kind of clinical populations we are talking about. But thank you.
what I think is that your work well about all your what really your career. And I have no particular quality for the internet during the present year, but what I want to reach is every success in your professional. And I think it's about thanks.
This subject was very good. And we were many people understanding, all the campus doing, every field they did, and doing, oh, we do and that's a myth to be like, and that's the best thing. You get harder. I mean, some very, let's say, down, 50 degrees, very good. So, you know, it's boy test. I mean, you have some insights from the work that's done. I know that you love open the food and stuff and
I think it's a very ambitious objective. It's already ambitious for the brain, for sure. For the cerebellum, it's even more ambitious as we have lower SNR and so on. To be honest, it was, among our objectives at some point when we first saw the possibility of having an instrument with more than 2 wavelengths. So we hypothesized to measure it. Unfortunately, this was not possible, but I think at the moment with the technology that is available in the market, let's say outside the market, this is probably mostly reachable by using broadband in-ears experiment in in-ears devices. So yeah, it never hurts to try, but of course we have to be cautious about it. It's a very ambitious. This was already ambitious, and measuring also cytochrome is an even more ambitious project, but it never hurts to try. Yeah. Thank you.
how you guys want me and young
Thank you a lot. Yes, this is a very good question. Who am I? And yeah, the truth is that so my background is biomedical engineering. I have bachelor and master in biomedical engineering. And I used to complain, I still complain sometimes because I feel that we say in Italian, we are not meat, not fish. We know a bit of everything and we are not real expert in anything. Like today I was really afraid of math questions and physics. Dramatic, no, I'm joking. And but it's the truth is that yeah, I feel for sure very much more confident on the signal processing and data analysis in general, also with statistics. I learned a lot during these years. But the skills I developed recently also, especially during the secondment in Canada, are experimental skills, which sometimes we don't mention enough. We talk about data, we don't talk where the data comes from. There are people behind it. And these are very precious skills, and it's difficult to find people. It was already difficult to find people, technicians that, especially for example, gluing ears. This is not something standard that they teach you at school, even if you are an EG technician or something. So I think these are skills that I could spend in the future if I still want to continue on the experimental part. And yeah, probably I feel I should learn more and should have a stronger background in neuropsychology, in the clinical part. But this is also why in our team we have a neurological expert. So yeah, so I'm a bit of an hybrid, let's say. Yes. Okay, just 1 second.
So we try to
Bon, je crois que la suite se fait sans nous, hein, c'est ça?
Ah, okay. We don't hear.
Okay, now.
Ah non, c'est bon, on entend.
Okay. Everything's fine?
Yeah.
Okay, what should I do?
Look at us. So, Giulia, you have addressed a very challenging topic. And the jury has found that you made a very clear presentation while introducing a lot of different subjects from different disciplines in a very didactic and pedagogical way. You have a lot of different questions from the members of the jury, and you have answered very clearly and with very convincing arguments. So all of these demonstrate to the jury that you have an initiative maturity and a deep understanding of all the aspects of your multidisciplinary subject. One encouragement for the jury is to publish your work, which is very original and innovative, and which very surely will become a reference in the community. So, at the unanimity, the jury has decided to give you the degree of the doctor of the University of Italy. And the jury has presented you to get the yearly PhD prize for the doctorate. Congratulations.
No, people are putting, you're supposed to read. No, first, you say that on active dialogue.
Okay. In the presence of my peers, with the completion of my doctorate in automatic traitement du signal et des images, in my quest for knowledge, I have carried out demanding research, demonstrating intellectual rigor, ethical reflection, and respect for the principle of research integrity. As I pursue my professional career, whatever my chosen field, I pledge to the greatest of my ability to continue to maintain integrity in my relationship to knowledge, in my method and in my research.
Right. Bravo.
Thank you. If I may, I have prepared a few slides. This time less technical, maybe more pleasant for the jury, for the audience. So let's see. The computer is almost crashing now that everything has finished. I understand it. Yes.
I will leave very soon, Giulia. I have my talk starting soon, so.
Yeah, sorry, Christophe. It would be very brief if I succeed to
Try to see your slide and then I go. Congratulations again. It's a great job.
No, PowerPoint doesn't want to start. I think it's the end, but
Okay. So I'll go. Bye-bye, everyone.
thank you, Christophe. Thank you for everything.
Congratulations, Giulia. Bye-bye to everyone.
Thank you.
Would be happy to be there, but next time. Bye.
You're welcome. All right.
Yeah, I'm rushing. I have
You close the
2 hours to teach now. Bye.
No,
Ciao.
I think.
Bye.
Caroline. Okay, I think time is running out. So I was supposed to present some thank you slides with some funny pictures, but it's not gonna happen. The file disappeared from my computer and PowerPoint is not opening, so you will have to be happy with my oral thank you. Sorry. But okay, so first slide. So for the people who read my thesis, I put a quote at the beginning of each chapter, and for the slides, I asked ChatGPT to write the quote for the slides, to write a quote for the end of the PhD. And they said that PhD is not only research, but is the survival of an existential crisis. So, this time we agree with ChatGPT. And then I wanted to say that for sure, there were many people who said like this journey was difficult. Why are you doing it? But I'm a very determined person, and when I like something, I'm convinced about something, I still go to pursue it. That's why today I came with a yellow blazer, even if everyone told me it was not appropriate, and you have a working highlighter in this room. But okay. And so want to thank, first of all, Dr. Elise Bagnier is on Zoom, Dr. Nadez Roche-Labarre for accepting to review the manuscript with much enthusiasm, for providing a very punctual feedback, and for spending their time and their attention to review it. Very much appreciated. Then I want to thank Professor Guilherme Wood, Professor Jean-Marc Lina, who came on purpose from Austria and from Canada to be here, to be part of the jury. Thanks a lot for reading the thesis, for extending this. Thank you, Professor Xavier Decombe, for accepting also with pleasure to be president of the jury. And then thank you to my three supervisors for accompanying me during this journey, which was not easy. Thank you to Jérôme, which believed in this project from the very beginning. He was the one that first convinced me that it was possible to measure cerebellar at years. So thank you for supervising me, for being very present on a daily basis, and for teaching me that research is taking risks. This is very important. Thank you, Marie-Noëlle, for being very gentle, and I'm not crying, for being very passionate about your work, for thanks a lot for your help and your supervision and everything. Thank you, Olivier, also for being support this project from the beginning, for being very kind in this academic world, which we know is not very easy. Then thank you to Professor Stefano Emanuele, not only for being in the jury, but for working together, because he's the one who introduced me in the fMRI world. Thank you really a lot for all your help, experimental and practical one. Thank you to Christophe, which unfortunately has to leave, for welcoming me in Montreal, really as one of the team. And I want to thank all my colleagues in Montreal, which are, this is getting difficult, which are not only colleagues, but friends, because they helped me a lot. It was very, very tough. Okay, I'm going to be fast because it's getting very difficult. Thank you to all the friends and colleagues that are here today, that are in Nice in the lab, for bearing all my complaints. Or I know I can be a difficult person sometimes, but thank you for all the good and beautiful memories that you gave me during these years. This were really unforgettable. Really special thanks to all of you also for helping me today with everything. Then thank you for who came from Italy on purpose to be here today, and also who couldn't. Thank you to my lifelong friends that we did school together, and they're here today. We celebrated everything together, from high school, bachelor, master, we're always together. And then the last thanks goes to my family and to my boyfriend that are a constant source of support for me. In all the difficult times, I wouldn't be there if it was not for them. Okay, it's becoming pathetic. Let's go to drink and to eat. Thank you, everyone. Thank you, everyone, for being here also online.